let's continue. Now, we've said that metaphase is the stage that comes after prophase. Therefore, since we are discussing on meiotic division, that phase is denoted by metaphase I. And remember, this is the shortest phase, as the tetrads align on the metaphase plane, or the homologous chromosomes align or they arrange themselves at the equatorial plane. And then another key event during this phase, which can lead to variation, is a concept that we call independent assortment. Remember, in Form 4, we discussed this as one of the causes of variation that makes individuals not look alike, despite the fact that they are born of the same parents. Now, as you can be able to see in this diagram, during metaphase I, the bivalents are orienting themselves along the equatorial plane, or the homologous chromosomes align themselves at the equatorial plane. And number 2, again, the spinal fibers or spinal apparatus, they lengthen. Actually, it is important to note that the spinal apparatus form during prophase I, then they lengthen at metaphase I. They lengthen at metaphase I, and once they lengthen, they attach themselves to the centromere. They attach themselves to the centromere, specifically at parts that we refer to as kinetochore. So these are the stages, or these are the behaviors of the cell at metaphase I. Now let's look at Anaphase I. During Anaphase I, homologous chromosomes separate and moves towards the opposite poles, and then the sister chromatids remain attached at their centromeres. The sister chromatids remain attached at their centromeres, can be illustrated using this diagram. So you'll be able to see here that the bivalents or homologous chromosomes are being pulled to the opposite pole. Remember, this is a centriole, and then from the centrioles there emanates the spinal apparatus, which attaches themselves onto the centromere, and then they are orienting or migrating to the opposite poles. The pair migrate to opposite poles. So when what makes them migrate to the opposite poles? It is believed that as a result of pulling of the spinal apparatus, they pull with them the homologous chromosomes that they have attached to at the points of the centromere. Therefore, the reason as to why the homologous chromosomes or the bivalents are pulling apart is as a result of the shortening of the spinal fibers. Again, it is important to say here that during prophase, the spinal fibers form. At metaphase, they lengthen, and here at anaphase, they shorten. They shorten, as illustrated. The last phase is the telophase. Telophase, which actually is the final phase that shows that the process of division has taken place. Now, in this case, each pole has a half the number of chromosomes. Each pole has a half the number of chromosomes. And then the process of cytokinesis occurs. Cytokinesis is the process of division that involves the cell membrane together with the cytoplasm. And the two haploid daughter cells are formed as illustrated in this diagram here. So you can be able to see from one single cell, there is the process of cytokinesis. Remember, the cell membrane constricting at this point in readiness to fully separate as observed in this second phase. So that means that during telophase, the process of first meiotic division has reached its peak. This is the behavior of the cell at late telophase. The behavior of the cell at late telophase. Remember, there is a new nuclear membrane that forms, This is not a cell membrane. This is a new nuclear membrane that forms, and then it bounds the chromosomes. So during a late telophase, they will not appear as chromosomes, but chromatin materials. So we shall look at meiosis II in our next lesson, and then after discussing the second meiotic division, we'll be able to differentiate between meiosis and mitosis, and of course look at the significance for each. Thank you.